Good morning, everyone. Today we are going to cover the final part of animal nutrition, which is topic 5.6, assimilation in the liver. Before we move on, let's do a recap session from our previous session, which is about human teeth. The learning objective was to identify the different types of human teeth and describe their structure and functions. We also needed to talk about the causes of dental decay and describe how to properly take care of your teeth. The human teeth is involved in the mechanical digestion, which is the first part of the digestive system. And you have two sets of teeth in your lifetime, which is the milk teeth. This lasts for about 10 to 12 years, and you have about 28 of them. And you also have permanent teeth. So after about 12 years old, the permanent teeth will then push out the milk teeth. And after 17 years of age, the wisdom teeth will grow, giving you a total of 32. There are different types of teeth, and they have different functions and also shapes and characteristics. So we have the incisors. There's a total of 8, 4 in the upper jaw and 4 in the lower jaw. The characteristic is very sharp, spade-like. Spade over here, this part that looks like a spade. The function is to bite and cut food, and the root type is single-rooted. Second type is the canine. You have a total about 4, 2 upper and 2 lower. The characteristic is that it's conical shape and very sharp. Conical means that it looks like a cone. The function of the canine is for biting, cutting, and tearing up food. And the root type, you can see here, it is single-rooted. Next, we have the premolar. This comes before the molars. You can see over here, the grayish part, those are the premolars, and the green color here, the last part, are the molars. Premolars, you have a total of 8, 4 upper and 4 lower. The characteristic is that the surfaces have about 2 projections, aka cusps, and they are used for crushing and grinding food. Root type is double-rooted. Lastly, we have the molars. There's a total of about 12, 6 upper and 6 lower. The characteristic is that it's about square shape and has about 4 cusps, so 1, 2, 3, 4. And this has a function of crushing and grinding food. Root type, mainly double-rooted. Sometimes they do have triple-rooted as well. Next up is tooth decay. The first stage of tooth decay is when food, especially sugary food, gets stuck in between your teeth. Then after that, bacteria will settle, using the sugar for their metabolism. So the bacteria will then secrete lactic acid, which dissolves the enamel, which is the outer layer, the protective layer of the tooth. And this causes a cavity, aka the hole here. And this develops and attracts more bacteria. Decay then reaches the living part of the tooth, which is the dentin, which is right inside here, which contains a lot of nerve ending, and this is where your toothache comes in. And when the decay reaches the pulp, which is this part right here, the reddish color, this can cause abscess, which means that it's kind of like, in Malay we say nanah, so it is very, very painful. Tooth decay also involves these two things, which is a plaque, and this happens when you have unclean teeth. The mix of saliva, cheek cells, and bacteria will collect at the neck of the tooth. So that means around here. So right in between your teeth. This deposit is called the plaque. Acids then is released by bacteria and damages the enamel, and because it's in between, so the plaque is in between the teeth, it actually is more in contact to the gums, so it can cause gum disease. But if it is not removed, so if this plaque is not removed, it can be hardened, and when it hardens, you call that tartar. Tartar protects the bacteria trapped in between the tartar and the tooth, also the gums. So this hardened plaque will then allow the teeth to decay without interference. This means that even if you brush your teeth, it's going to be really difficult to remove it because it's very, very hard. So how do we take care of our teeth? Firstly, do not eat starchy or sweet food before bed. Secondly, brush your teeth last at night and first thing in the morning to remove the plaque. Oh, I keep on pronouncing it wrong. Use dental floss regularly to remove fragments of food between the teeth, and use toothpaste that contains fluoride, bactericide, and is alkaline. You also should visit the dentist regularly. Now let's move on to the first part about the liver and assimilation, which is the hepatic portal vein. But before that, I want to show you guys a 3D diagram of the liver. So here we have anatomylearning.com, and it shows like 3D images of basically the human body. And right there, that's the diaphragm, and underneath the diaphragm is actually your liver. You can see down here that the liver is connected to the gallbladder. And also there are these two colored tubes. This one, this really, really big one colored blue, is called the hepatic portal vein. And it actually comes from somewhere down here. There's also this little red tube, and this is called the hepatic artery. Arteries are blood vessels that carry oxygenated blood from the heart, and this actually supplies the liver with a lot of oxygen. Let's have a look at another 3D diagram of the liver. You can see here, right there, that is called the inferior vena cava. And you can see that it's really big. Inferior vena cava, so vena cava actually returns deoxygenated blood back to the heart. So let's try to kind of remove the stomach because it's kind of really covering a lot of stuff there. Let's remove the pancreas. Okay, let's see there here. So we have the inferior vena cava behind the hepatic portal vein. The hepatic portal vein is actually connected to, if you can see right here, it's actually almost connected to, but it should be connected to this part right here, which is the small intestine. So the duodenum is this C-shaped, U-shaped part of the small intestine. And the small intestine, the whole of it, is actually connected to the hepatic portal vein. The hepatic portal vein also comes up into the liver. So we're going to talk about what it actually transports. So let's have a look at this one right here. It gives you a bit more of a translucent image, so you can see the blood vessels and how it connects. So let's remove this segment right here, move it away, and you can see there are different types of blood vessels. So you have right here the portal vein, which is connected to the hepatic portal vein, and then you have this color right here, the brighter blue color. So the darker blue refers to the hepatic portal vein, and the lighter blue refers to the hepatic vein. The hepatic vein is actually connected to the vena cava, the inferior vena cava, whereas this hepatic portal vein is connected to the small intestine, the duodenum that you saw just now. It also is mainly connected to the ileum and also the large intestine. And here we have this little red part here, which is the hepatic artery. So now that you kind of have seen the 3D image, so we're going to look at the 2D image as well. So let's move back to our slides. So the hepatic portal vein. The learning objective 5R states that you need to state the function of the hepatic portal vein as the route taken by most of the food absorbed from the small intestine. So what the hepatic portal vein does is that it actually transports most of the food that is absorbed in the small intestine and transports it to the liver. So let's give you guys an overview of basically everything. So you have a liver right here, and just now you have the red blood vessel, and this is called the hepatic artery. Arteries are all the blood vessels that carry oxygenated blood away from the heart. So you have the heart right here, and you have the left side. No, this is the right side of the heart, and this is your left side of the heart. Whenever we're looking at the heart, we're pretending like we're seeing a patient lying down. So this would be the right hand side, and this would be the left hand side. So this one right here, this main artery that brings blood away from the heart is called the aorta. Aorta transports oxygenated blood to all parts of the body, including the liver. And the main function of this is so that it can transport oxygen for the liver so that the liver can do its main functions. There's another blood vessel that we saw, the really big one at the back, and that one is called the hepatic vein. Hepatic vein is the blood vessel that will be connected to the inferior vena cava, which is right here. This is the superior and inferior. Inferior vena cava carries blood, or brings back deoxygenated blood back to the heart. So the inferior one actually transports the blood that's like below the heart, like the body, that part. So the hepatic vein will transport the deoxygenated blood back to the heart. So this means that the blood from the liver will then move out from the liver and into the vena cava. And then after that, it's going to go through the cycle, the circulation. We also looked at another blood vessel, which is called the hepatic portal vein. And this is actually connected to the small intestine and also the large intestine, but we're going to focus on the small intestine first. So in the small intestine, if you remember, there is folded walls, and on those folded walls you have lining, even more folds, called the villus. And if you have a lot of them, like here there's 3, you call that the villi. And the villi is made up of the epithelium, which is epithelial cells, and inside them you have the blood capillaries and lacteal. So the blood capillaries will then end up to make this blue part right here, which is the vein. And what happens is that sugars and amino acids will be absorbed into the blood capillaries and will be transported through this vein. And this vein will then be connected to the hepatic portal vein. So all of those sugars and amino acids will actually be carried to the hepatic portal vein. Also connected to the hepatic portal vein is the large intestine, and in here the main function of the large intestine is to absorb water and minerals like calcium and iron. And what happens is that the absorbed water and the minerals will also be carried towards the hepatic portal vein. And where does this end up? It's going to go up into the liver. So the hepatic portal vein actually carries or transports sugars like glucose, amino acids, water, and mineral salts from the digestive system, which is the small intestine and large intestine, towards the liver. What does the liver do with that is that assimilation. Assimilation of sugars and amino acids will happen in the liver. Now let's look at the learning objectives of 5S, which is to state that large molecules are synthesized. Synthesized means that it is made from smaller basic units. For example, glycogen, which is a really large polysaccharide, it is made from glucose. And protein, which is a large molecule as well, it's made up of tiny little bits of molecules called amino acids. Lipids is made up of glycerol and fatty acids. And we need to learn about the role of the liver in terms of metabolism of glucose and amino acids, and also talk about the role of fat as a storage substance, but we're going to move on to this next lesson because I want to focus mainly on the liver today. Lastly, the formation of urea and the breakdown of alcohol that occurs in the liver. So what is assimilation? Assimilation happens when we have small food substances that is absorbed from our, usually the small intestine and also the large intestine. And these small food substances actually are needed to build up larger molecules that are needed by the body. Assimilation is the transport and utilization. Utilization means to make use of the absorbed food to build them into larger molecules needed by the body. Assimilation mainly involves two substances, which is glucose and amino acids. So let's look into the assimilation of glucose first. Glucose can be used in the process called respiration, which is glucose plus oxygen, and then you're going to get carbon dioxide, water, and a lot of energy in the form of ATP. Glucose, when you have excess, so after all the respiration has happened and you get still excess glucose, you are going to process that as well. So what happens is that when you use up glucose for respiration, the hepatic artery actually carries oxygen to the liver by transporting oxygenated blood. Remember that red blood cells carry oxygen. This oxygen will then react with glucose in the mitochondria to produce energy during cellular respiration. So the main products of respiration is high amount of energy in the form of ATP, carbon dioxide, and water. When you have excess glucose, so you already used up that glucose, but you still have extra, it needs to be stored. So what happens is that the pancreas will release this hormone called insulin. And this insulin will be transported to the liver. And under the effect of insulin, the liver and muscles will convert the glucose into a bigger storage form of sugar, which is glycogen. Now let's look into amino acids. Firstly, why is amino acid really important? Amino acids are used in the cell for various purposes. Firstly, to build up proteins for cell growth, and secondly, to make enzymes. Amino acids and proteins are actually never stored, so what happens to the rest of it if you eat too much protein? Any excess to our needs at the time will be broken down in the liver by this process called deamination. Amino acids, you have quite a lot of it if you eat a lot of protein, and its uses, you can use it up or you have extra. It's used to build proteins, and it's also used for cell growth. So proteins are actually found in all parts of the cell, like the mitochondria, and even in the cell membrane, like the transport proteins in between here. Amino acids are also used up to make enzymes and hormones, like enzymes like your digestive enzymes and hormones like insulin. But if you have extra, then your liver will carry out this process called deamination. So your amino acids is actually going to be broken down into two groups. The first one is called the amino group, and the second one is the carbon skeleton. So if you look at the structure of amino acid right here, this is just one amino acid. You can see that it's made up of carbon, nitrogen, hydrogen, oxygen. And this part right here within nitrogen is called the amino group. And the rest right here is called the carbon skeleton. So after deamination, the amino group will be converted into urea, which contains a lot of nitrogen, and it's actually quite toxic for your body. So here we can see the nitrogen and hydrogen. So it's all made up here from this part of the amino acid. This urea will also be passed to the kidney for excretion. So urea will only be removed at the kidney. It's not going to be removed from the liver. The liver only carries urea. So urea is actually added to the liver, and then it's going to be transported to the heart, and the heart will then transport it back to the kidney, and that's where excretion occurs. That's when it's going to be removed in the form of urine. What happens to the rest of it? This one here is that it's going to be changed. So the carbon skeleton will be changed into glucose and stored as glycogen in the liver. So just to summarize, the liver has a major role in terms of glucose and amino acid metabolism. Metabolism is the sum of chemical reactions that happens in your body. And the liver involves itself in the regulation of blood glucose levels. Blood glucose level, or any blood level, refers to the level of that thing inside the blood. So glucose is actually dissolved in the plasma part of your blood, and the levels actually fluctuate depending on if you have eaten or if you're fasting, for example. Another thing is that metabolism also includes the regulation of amino acids, and amino acid is also dissolved in the plasma part of your blood. In terms of glucose level, the liver will actually react if there is high level of glucose in the blood and if there is low glucose in the blood. What happens if it's too high in the blood? And if there's excess of amino acid as well. So in the case of high glucose level in the blood, the glucose will be converted to glycogen and be stored in the liver. In terms of low glucose, so this happens when you're fasting, glycogen, so the stored glucose, will be converted into free glucose that can be used for your cells. So this one, glycogen stored in the liver, so glucose to glycogen, happens right after you eat. And this part right here happens when you're fasting or when you're hungry, when you need to eat. So when we have excess of amino acid, we have this process called deamination. So this is the breakdown of the amino acid into urea, and urea will be removed only at the kidney, and sometimes I think involves in sweat as well, so that is also excretion. The liver also has another important role in your body, which is called detoxification. So whenever people talk about detox tea and detox food, actually your liver does all the detoxing for you. Detoxification means to remove and break down any poison or toxins from the blood. So one poison is actually alcohol, because it is not good for your body. Liver can remove small quantities of alcohol on a regular basis. But if there is high level of alcohol in the blood, this can eventually lead to this disease called cirrhosis. So here's the healthy liver, and you have the cirrhotic liver. Liver cirrhosis just means that when the liver has been doing so much job of detoxification, the cells actually become scarred, and then when they heal, they become hardened. So this is what happens when people consume way too much alcohol. And that is the end of today. We're going to talk about what happens to fat in our next lesson. So hope you guys have a good day, and I'll see you guys next time.